Hello all. So I want to give you about a 10-minute introduction. There will be more to come after watching this video. I'll send you to our website at the end of this PowerPoint that will allow you to review these concepts and more. These are just very basic introduction. It should take a total of about 15 minutes of your life. And then I will have the tutorial posted for the week. So I'm going to share my screen and go through a very brief PowerPoint. So introduction to genome projects. You are not going to complete a genome project. That's not what I mean by projects. You are going to, the end result is to annotate a gene or locate structures of a gene. How do we do that? So we're going to start off by defining what a genome project is and what's involved. What is the difference between, say, gene sequencing, which is what we used to refer to as a genome project, like the Human Genome Project. We had a quote-unquote end to the Human Genome Project a couple of years earlier than targeted for. I believe it was in 2001. And what that really did was it sequenced the entire human genome, and we were able to identify certain families of genes, hundreds, thousands of them, but that really did not conclude. That really just opened up a whole new book to other information. So how and where is information that we do in the sequencing and the annotating, kind of like you would annotate in the side of your pages of a book you're reading, talking about what's happening there. So we do the same thing with genes. How and where is this information that we learn produced by genome projects? Where is it stored? Keeping in mind, of course, we're talking about chromosomes. Now we can see chromosomes with a powerful enough microscope, and we can do the karyotypes to count the chromosomes, but it has no bearing on what this actual sequence is. So trying to find out what is that sequence of DNA for any given chromosomes and where are those genes at, that's closer to what the research societies do, right? So you already know that DNA is a series of nucleotides that are complementary to each other. So A to T, T to A, G to C, C to G, and so forth. And you also know there's a start codon, ATG, which will code for a UAC, mRNA. U replaces T, let's not forget our basics. And then stop is TAA or AUU, or the RNA uracil replaces thymine. So these things exist. So you could, and there are algorithms that will scan a sequence. So first we have to have sequenced the DNA, and then we can look for these various elements. And so we could tell a program, find all the ATGs, and that's going to be our clue as to how many genes are there, right? Kind of, not quite as easy as that, but just for the sake of the example right now, yes, that's what you would do. You would type in your parameters for the program and say, find all the ATGs. And you could even say, find the ATGs that are upstream 5' of a TAA site. And then it would just give you the ATGs that are upstream of a TAA. And you would say, oh, boom, right there, right in the middle. This is where the gene is then. And this could be any length of DNA. It's a very simplified way of looking at it, but that is pretty much what you're doing. You're just plugging in parameters, or you're looking for these things, and you got to tell the computer what to look for in this. So what you get initially, what you're doing is sequencing, right? So this is just somewhere in the genome, millions and sometimes billions, depending on the genome, right, of nucleotides. And a computer readout, after doing some of the biotech methods we've referred to, we haven't talked in great depth about this, but we've referred to sequencing and other PCR and chromosome walking and different ways to get a sequence. And that's all you have, those sequence. What do you do with that? Is that whole thing needed? Is just a part of it needed? In some of the papers we've reviewed, we know that, when we talked about phytochrome protein, there was a LOVE1 and a LOVE2. We mutated the LOVE1, nothing happened. Mutated LOVE2, and we lost function. So what part of this sequence is most important or needs to be conserved? and what part, if it mutated, would be fine. So we need to learn that, and this is what we call bioinformatics. Find out the information of this molecule. After we sequence it, sometimes, not always, a good thing to do, and what some people are interested in, is mapping the genome. So for a given species of interest, we learn there are model specimen that we have the gene sequence to, and we can learn a lot about our own physiology and about plant physiology. And we can begin to select for traits if we know where the gene for a particular trait is located. So for some trait, let's just say resistance to glyphosate. And we find out that all these genes and through other biotech methods play a role. And so we know certain of these sequences have a role. And notice these represent chromosomes, right? And megabases. So 5,000 or 10,000. So you have this represents a chromosome bars, and different chromosomes are different sizes, and there's locations. So how do we learn this stuff? It's clear that a more demanding definition of a finished genome requires extensive annotation of the assembled chromosome sequences in addition to the mere sequence report. So what again is it to annotate? Well, it's the process of finding genes within a genome and defining the structures or the functions of those genes. So structures might include the open reading frames, could include exons, introns, regulatory regions, of which there are many, the Tata box, the cat box, the poly A tail, introns, exons. So the functions would include roles of gene products. So what is a gene product? Typically a protein, we think, but also could be mRNA, tRNA, rRNA, and other regulatory features of genes. So locating these things and then testing them out in the lab, that's what annotation attempts to do. Here's an example of an annotation. What's actually here is not important, but this is representing a sequence of DNA. And someone who studies this has learned then, and maybe it's a series of labs or people, somebody probably discovered this retrotransposon, right? And saw that they vary in number and size throughout the genome, and lo and behold, here is one. And so we can use, that's good information. That might throw off how these genes are read, right? They used a triangle to denote mites, often occurring genes, promoters, and matrix attachments. They use all these black bars to represent the exonic regions. Oh, I'm sorry, black. And the white ones are the intronic regions, non-coding for any RNA or protein. Okay. The asterisk is a simple sequence repeat, often occurring intragenically, which simply means in between genes. And we use those oftentimes as markers because they tend to stay in a location and they mutate. We can use them to distinguish one species within a population from another. If you have enough of those SSRs, we try to find, that's what we do in our lab, right? And the more SSRs we have, the more solid conclusions we can make about who and what traits and parents that individual belongs to. So these are markers along the way. So all I want to show here, this gene is read this way, this one's read this way, this way. So this is the bottom strand, it's the top strands. So all this conveys information. You leave a key for your reader, and you can annotate. So an example, genome project then includes both the sequencing stage and an annotation stage. An example of my work, I don't do sequencing, although sometimes we have to, we'll send it off. We're usually working with organisms from which there's already a sequence or some amount of the sequence. And we're interested in finding a location for where our sequence is in the genome so that we can use it and look for it in future studies using, of course, computers. Let me repeat that. So, for instance, in the grapes or black walnuts I work with, I don't need to sequence. Others already have, but I am interested in a particular gene of interest, right? And if we find that then, so through breeding and crossing and having a mapping population of many individuals, there's going to be some commonalities between their traits and their genomes, and we can begin to look for locations within the genome where these gene sequences reside. And if we know that by a marker, for instance, or by the gene, then we can begin to look for those traits in our individuals early on in their life instead of later. So here's an example: detection and quantification of how much, right, of Roundup Ready soybean residues in sausage samples by conventional and real-time PCR. So these people use real-time PCR. This is the sequence of the primer. So if you wanted to do the same research and look for these proteins, this is what you would order from the supply company. You would order a 5' to 3', GCCTCTACT. This is exactly what you would write down or type into the order form. And these are called oligos. There are a few nucleotides long, about 20, usually 18 to 21. And these are the primers that would be laid down for your PCR. And over here, it's telling you the amplicon size. So in PCR, you have these primers. When you run electrophoresis after your PCR, you should get about 118 base pairs, 180 base pairs, 172 base pairs. So this is the full length of the DNA strand in relation to these targets. Okay, I know we don't understand what all these are, but you can tell they're like genes. They're parts of the genome for something. And here, this was being used as a method to detect and see if that Roundup Ready gene was being, what's the fate of that? This has bearing on GMOs, right? So we have a genetically modified soybean. It's been treated and such that it's Roundup Ready resistant. So now you can spray Roundup everywhere, the glyphosate, and you don't have to worry about killing your soybeans because the soybean is resistant. Well, we feed that soybean, we harvest it, give it to the pigs. The pigs are then harvested and sausage. And so now we're looking for and detecting any residues of that soybean resistance gene in our diet. And how that travels through a living cycle or an ecosystem is pretty interesting. Now, whether that's actually bad or good, I'm not saying. Just saying this is one application of knowing a sequence, in this case, the primers, and also finding out just how big that gene is. So if you have all this information, like let's go back to this real quick. How do we know where we store this information? So community databases or data warehouses, and these are very specific to the organisms and we test. Now, a lot of these, especially in mice, would also be found on in databases such as NCBI. They have a lot of animals, actually, organisms are there, model organisms. But these are different databases: Wormbase, Maze, GDB, SpudDB. So if your whole career is corn improvement or rice or grape, then you're probably going to belong to a society or and have subscription or at least access to these databases. And I'll click on one. Click on. Oh, I don't have it active. That's all right. You can click on them. I'll post this, and it'll take you straight to the home page of each of these sites. I just copy and pasted this off of their website and put them here. All right. So that's the short of it. There's sequencing and there's annotating. Sequence is just knowing what nucleotides are there. Annotating is knowing the significance or importance or the function of those nucleotides. And ultimately, sometimes it's to map where those are at. Just depends on what questions you're trying to answer. And bioinformatics are the tools and the stronger, and there's all kinds of programs, so there's all kinds of algorithms that they use in order to hunt and find. We've already learned so many of them just in our class, and this is just an introduction. So what you're going to do here is you're going to click on Quick Review, and you're going to go to a website that looks like this. Now, this is Flash, and so it's going to be blocked for the probably. So you need to either approve it over here. If it has the little puzzle piece, click on that, and you will have to go to your settings and allow the pop-up or allow unblock Flash to do this. Okay, I already unblocked it so you can't see it anymore, but there's a puzzle piece here. I'm using Chrome. Sometimes it's something over here or drops down, but you'll have to allow it in order to go through it. This will take you 5 to 10 minutes, just depends on how fast you read and whatnot. So go through, slide by slide introduction. And then there'll be some quiz questions, 5 quiz questions. Make sure you spend some time here to learn what these things are. And this is just one database. Everybody else's will differ just a little bit, but just to give you a sample. All right, that's an introduction to genome projects. And that concludes this short.